Well, I think I'm just going to start with some introductory remarks. Good morning, everyone. It's a great pleasure and a distinct privilege to be here to welcome you to the first Munck-Pfefferkorn Prize Lecture. And as you know, the Munck-Pfefferkorn Prize Lecture Series was established to honor Drs. Alan Munck and Elmer Pfefferkorn. Both were consummate scientists. Alan Munck was the discoverer of the glucocorticoid receptor, and Dr. Pfefferkorn was a consummate parasitologist who did seminal work on the action and function of interferons. Both were beloved faculty in our medical school for over 40 years, and both consistently received the highest accolades for their teaching and mentoring. From medical students, from graduate students, from postdocs, and also from faculty that whom they have mentored. When the organizing committee asked if I would recommend a world-renowned scientist to kick off the Munck-Pfefferkorn lecture series, it took me less than the time it takes for a synaptic delay to actually sit down and email Dr. Tom Südhof. And, Tom, I'm just elated that you are here and accepted the invitation. Thank you very much. And I'm not going to say too much more because Brian is going to be here. Brian Lucart is going to be here to do the formal introduction. So, Brian.
All right. So thanks, Hermes. I'm also elated that we have Dr. Südhof here. And I'm extremely proud to be able to introduce the first Munck-Pfefferkorn lecture. And so I think one of the important founding principles of this lecture is important mentorship, good mentorship, as Munck and Pfefferkorn were. And I think that's also been important in bringing us the scientific teachings of Dr. Südhof. So Dr. Südhof obtained his MD in Göttingen, and then after that, he came to UT Southwestern in Texas to do a postdoctoral fellowship with Drs. Brown and Goldstein, where he cloned the LDL receptor. And at that point in his career, he was considering going and becoming a physician and not staying in science. So, and as the story that Dr. Brown tells, he basically told Tom that if he left science, it would be a great tragic loss for science. And so, between that and offering him a laboratory at Southwestern, we were lucky enough to have Tom at UT Southwestern. So I was a student there. I was fortunate enough to work in Tom's lab and to attend classes on synapses that he taught. And one thing about Tom is that we have works in progress at UT Southwestern, and Tom was always there to give his opinion. He had a strong reputation of not ever holding back his opinion. And I think that sort of rigorous opinions of his are a fundamental tenet for being able to defend yourself amongst the greater scientific world. And so in learning how to do that and in paying attention to the questions that Tom really liked to discuss in classes, it actually shaped my career to a great degree. And so I'm very excited to be able to introduce you here to hear him talk about the molecular dissection of synapse formation and specification by Neurexins and company. And I hope that you're able to be as inspired today as I was as a student around Tom. So with that, thanks.
Good morning, everybody. Thank you for this wonderful introduction. So nice to hear oneself being characterized as not holding back. It is an honor for me to be here. I haven't been at Dartmouth for a long time. the last time I was here at the invitation of Bill Wigner, who was sitting there, I was mostly interested in vesicle traffic. But even at that time, a considerable part of my laboratory focused on how synapses are formed and specified. And over the years, that interest has sort of slowly shifted. And what I want to talk about today is our work, as mentioned by the title, ongoing work on how synapses are formed and specified, focusing on one particular family of proteins, Neurexins. But before I actually tell you, sort of lead you into this whole subject, let me just remind you, for those of you who don't know, maybe we should have Donald Trump here or somebody like this, that the brain is very complex. And it's complex just based on its sheer size. What you see here is obviously dead brain. And in the brain, the human brain, there's about 10 to the 12 neurons, give or take a tenfold difference. At each of these neurons forms thousands of synapses to connect these neurons into vast networks of communicating cells. When you compare that with the size of the human genome, you have to admit that the genome is simple in that comparison. It is actually many, many orders of magnitude smaller than the number of synapses in the brain that form the network, that form the wiring diagram, the wiring pattern of the brain. And as you all know, we actually don't understand exactly how the genome works. So clearly, just by sheer size, understanding the brain is arguably one of the most interesting and most important challenges we face in biology. Also, because, after all, the brain is important, I would hope, and, and there's a lot of diseases of the brain. Given the situation, it's not surprising that people are arguing about how to approach the understanding of the brain. I can't help showing you this news and views write-up from Science recently, half a year ago, that reports on a meeting that tries to discuss how to approach the brain, how to understand the brain. And in particular, I want to point out this one section here where you can see a discussion of suggestions of what's the most important thing, like, for example, to cure a single illness or disorder. Now, obviously, we all would love to cure illnesses and disorders. A single sounds not very ambitious, but it turns out that as regards the major diseases of the brain, in fact, it is very ambitious, because none of the major diseases at this point are really treatable very well, maybe except for stroke. Another suggestion was to make a detailed map of brain connections, which means basically establish what neuron actually contacts what other one, which, as you can imagine, based on the size of the brain, is a massive undertaking. Another one is to crack, for the first time, a single complex behavior. And I think all of those goals are clearly interesting. You know, people always want to work on things like consciousness because people are interested in their own consciousness. Yeah, and in fact, last night at dinner we discussed consciousness. But the question is this actually possible, or isn't this all kind of mind play? Because what I would like to argue, and what my own presentation is based on, is the notion that understanding the brain actually requires understanding its components. It's understanding really fundamental issues like how does a nerve cell actually work? What is the cell biology of the nerve cell? How do synapses work? How do nerve cells integrate information? Just very fundamental, basic problems that are completely unsolved in neuroscience. In fact, we don't actually even know how a nerve cell becomes a nerve cell in the first place. When you look at the fundamental unit that composes the brain functionally, it is generally considered to be the circuit. And the circuit is basically any set of neurons that are connected by synapses, as schematically shown here, for a simple circuit, which is a typical, what's called a feed-forward inhibitory circuit, where you have one excitatory neuron that excites another excitatory neuron, but at the same time excites an inhibitory neuron, which then inhibits this second excitatory neuron. So when you look at this, what's obvious is that the key part of that neuron circuit are the synapses, because the synapses make the circuit. Without synapses, there's no circuit. But understanding even this fundamentally simple connection is actually very difficult if you don't understand its individual components. And let me tell you why. That is because at the synapse, information transfer is not linear. It's not like you have a digital signal that says yes or no. Synapses are themselves information processing units. And as a result, you can't actually predict whether a particular pattern of action potentials in the presynaptic neuron here elicits a particular postsynaptic signal in either one of these neurons, or from here, what did I do wrong here? Something. Oh, okay, or from here to here. You can't actually, even if you know such a circuit diagram, predict what the circuit does or how it reacts, what the input-output relations are. So what we really need is to understand the cellular properties of synapses, of neurons, fundamental building blocks. Otherwise, even if we had a map of the whole connectivity of the human brain with its 10 to the 16 synapses, we wouldn't be able to actually understand what that means. In fact, we've had a map of the connectivity of the worm, C. elegans, with only 302 neurons now for 30 years, and people still publish many, many papers on how this neuron or that neuron does this or that. So this is the fundamental problem. What I'm particularly interested in within that problem is how synapses specifically are formed and endowed with particular properties. The hypothesis that our work guides our work and that guides my presentation here today is that this process depends on interactions between the pre- and postsynaptic side, as shown here in this electron micrograph of a synapse, and that there's a bidirectional signaling that tells the synapse when to form and what kind of synapse to become, what properties to have, how to react plastically to certain input patterns, this kind of stuff. And so the goal of our work is to elucidate components of what this molecular machinery that dictates the establishment of synaptic connections and their properties. Now, in understanding the brain, in understanding how the brain works, one particular area that has made tremendous progress is genetics. And that is because of the enormous progress in sequencing. The cost for sequencing a human genome routinely will be less than $1,000 pretty soon, if it isn't already. It used to be, what, 3 billion only 15 years ago. It's amazing. It's unbelievable. As a result, that is the one area where progress has been made, I believe, in understanding neuropsychiatric disorders. As an example, autism, which is known to have a significant hereditary component, has now been studied in detail in terms of the genetic basis. And the results from such studies have been surprising, because everybody thought that given this hereditary component, there would be a few genes where mutations might cause the disorder. Instead, what has been discovered, as shown very, very exemplarily in this paper, which is only one of literally hundreds of papers that have been published, what has been discovered is that there's many different genes that have no apparent connection. This one described 71 risk loci, but people go from anywhere from 50 to 500. And so this has been a surprise, but also a conundrum. because in trying to understand how the disorder might arise, having a lot of different genes that have no apparent connection with each other is a problem, because you can't really conclude much. It's been tried to actually analyze these genes using some kind of systems or network analysis. This is actually a figure taken from the same paper where such approaches were used based on literature data that some undergraduate students in some major universities, such as this one, put together. What this leads to is these kinds of networks that you see here, where you can see that the various genes that were identified are connected by lines that are supposed to express some kind of relationship. This turns out to be useful, but only limited so. For example, just to key in on this part of this network, because these are genes I'm going to talk about, what you see here is that neurexins and neuroligands, which are what I'm going to talk about, are connected, and they are really connected because they bind to each other. So that fits. But you also have a transcription factor that clearly doesn't bind to neurexin. Thank God there's something in between, but it's only one. And you have all these kinds of things that don't really make much sense, but are still connected. So I'm telling you this as to illustrate what the problems are, why it's important to actually get down to the real molecular cellular biology and physiology of these units, neurons, synapses, whatever, because we really can't understand how this works. And my talk today, I want to illustrate how I believe we can make progress both in the basic understanding of how the brain is designed, as well as for possible translational understanding of how mutations in particular genes might predispose to certain disorders, by telling you about our work on neurexins and neuroligands. So this is what I'm going to talk about. Long introduction. Thank God we started on time. So I'm first going to introduce you to neurexins. I'm then going to very briefly tell you about one approach to try to model human neurexin 1 mutations in human neurons as a way to test whether these are actually physiologically or pathophysiologically relevant. I'm then going to tell you in a bigger section about the function of neurexin 3, which is the one we best understand, to illustrate to you how diverse these functions are. And finally, I'm going to tell you how we studied a neuroligand, which is a ligand for neurexins, as a way of figuring out how a particular mutation might actually predispose to behavioral abnormalities that are observed in autism. This is not a comprehensive review of the field. One reason is that at this point we are a little confused about the whole thing. We have bits and pieces. We don't have a whole view. Okay, so let me start with the beginning. I already mentioned the fundamental guiding principle of our work, which is the idea that synapse formation and specification are controlled by transsynaptic interactions between the pre- and postsynaptic neurons. Neurexins were the first transsynaptic signaling molecules that were discovered. They were discovered when I was at Southwestern, before Brian ever put a foot into the institution. When we were working on this beautiful spider that doesn't exist in either New Hampshire or Vermont, I'm sure, because it's a black widow spider and there's no deserts here. Black widow spiders are fascinating and beautiful creatures. They're wonderful. They're particular role models for females at some points, but maybe not around here. This is in California we live in a different life. But anyway, but they also have other uses besides being a role model. And that is that they produce a venom, and that venom has a toxin. That toxin is called alpha-latotoxin. Alpha-latotoxin binds to presynaptic terminals, and it triggers neurotransmitter release. Neurexins were identified as receptors for alpha-latotoxin. When we cloned them, we found that they are composed of a family of proteins that come in two principal forms: alpha neurexins that are longer, have a complex extracellular domain structure, beta neurexins that are shorter and just contain a single one of these LNS domains. beta neurexins are transcribed from the same genes as alpha neurexins from an internal promoter. There are 3 genes that encode alpha and beta neurexins in the human genome. They're subject to baroque, extensive alternative splicing, creating thousands of isoforms as demonstrated by packed biosequencing. And there have been many mutations that have been associated, actually hundreds of mutations, that have been found in patients with autism and schizophrenia and other neuropsychiatric disorders, and that predisposed to these disorders. The baroque alternative splicing of neurexins is something that interested us from the very beginning because it suggested that these molecules have the potential to encode information, to represent a recognition code. More recently, we have demonstrated that this alternative splicing does indeed occur in a manner that's specific to particular types of neurons, suggesting that that code actually exists. For example, in a recent set of experiments, we aspirated cytosol from identified neurons, interneurons in this case, PV positive and CCK positive interneurons in the hippocampus, and then we performed quantitative RT-PCR on single neurons for hundreds of genes, including various splice variants of neurexins. What we found as an illustration is that the neurexin 1 alpha is differentially expressed in these interneurons, whereas neurexin 1 beta is not. We then looked at the alternative splicing of just neurexin 1 for these 3 different sites of alternative splicing, SS2, 3, and 4, and looked at it by using primers that specifically differentiate between the versions where there's an insert and where there's no insert, as shown here. And when we looked at this, we found that for each one of these splice sites, the pattern was different between these 2 different types of interneurons, just as an illustration for this idea that extensive alternative splicing at different sites creates an information code that is present, differentially expressed in different types of neurons. And as an aside for those who are fond of RNA-seq, this kind of work cannot be done by RNA-seq because the depth of sequencing is never enough. It's interesting, for example, that alternative splicing is not digital in that these alternative splice sites are only partially used one way or another. It's not always in or out. It's always somewhere in between. With this information, what emerges then is that neurexins are displayed on the neuronal cell surface in many different forms. They look like receptors. They presumably have ligands, and it turns out they have many. Over the years, many have been characterized that bind to neurexins with nanomolar affinity. What this slide shows is just 4 of them. 4 that bind to both alpha and beta neurexins, and thus to the more C-terminal part of their extracellular sequences, and that bind with nanomolar affinities. What is interesting here is that these transcellular ligands that form junctions by binding to neurexins have nothing in common. There's no sequence motif that unites them. There's nothing like a neurexin binding site that you can actually identify by sequence. You have among these proteins a C1q domain protein that also binds to glutamate-like receptors. You have neuroligands, the first ligands identified. You have a leucine-rich repeat protein, and you have a GPCR. Very, very different proteins. However, apart from binding to neurexins, both to the alpha and beta form, what unites these ligands is that they're all also regulated by alternative splicing. They're regulated by alternative splicing in a sequence that is in both alpha and beta neurexins and alternatively spliced here, splice site number 4. And they're differentially regulated in such a manner that sometimes the ligand will only bind when there is actually an insert in splice site number 4. I don't seem to, this thing doesn't like me, that's for sure. When there is an insert, some only bind when there's no insert, and some bind with differential affinities with insert or without insert, depending on how the alternative splicing of the neuroligands in this case is. So what you have is an interaction network, a molecular network between different proteins whose nature is determined by alternative splicing, by the relative affinities of different isoforms, and by the relative expression of different genes. Among these different ligands. Ligands may be the most important neurexin ligands, first of all, because they're evolutionarily conserved, and second, because they are most likely the most abundant of these ligands. There's 4 evolutionarily conserved genes that encode neuroligands. Again, there's many mutations in neuroligands that have been associated with autism. And among these 4 different neuroligands, there are differential distributions and different properties that I won't go into at this point. So much for an introduction for Neurexins. Obviously, one of the key issues whenever a mutation is described in a gene that is supposed to be associated with a neuropsychiatric disorder is whether that mutation actually does anything. This issue comes up particularly with genes where mutations are very rare. In cases like Neurexins and neuroligands where hundreds of mutations have been described, one has relative confidence that they are actually meaningful. Nevertheless, we wanted to actually know whether a mutation, a heterozygous loss of function, in this case of Neurexin 1, which is the most frequently observed mutation in schizophrenia and autism among Neurexin mutations, whether this mutation had actually a functional consequence. And so what we did is we first looked at these mutations just as a way of sort of surveying the landscape. What you see here is a distribution of these mutations, which are invariable loss of function mutations that consist of deletions across the Neurexin 1 alpha and 1 beta gene, and you can see that there's many different mutations that cover the entire gene and thereby are predicted to be heterozygous loss of function mutations. We were then interested to figure out whether these mutations are actually meaningful. What was interesting about this patient observation was that these mutations seemed to be associated with a large variety of different disorders. There seemed to be no consistent clinical picture emerging that was associated with these mutations, leading us to hypothesize that maybe there's other factors, such as genetic background, that may play a role. So we wanted to actually test these mutations as well as other mutations in other genes that were associated with autism for their pathophysiological relevance. And to do this, we created human ES cells that contain conditional mutations in Neurexin 1, allowing heterozygous conditional deletion in a manner that when you create neurons, as shown here from these ES cells, you can actually introduce the mutation after induction or during induction of the neurons, thereby preventing the need to ever isolate mutant clones, which is the major problem in human ES cells or IPS cells. Not surprisingly, when you delete one of the 2 copies, one of the 2 alleles of Neurexin 1, you get less expression, as shown here. And when we analyzed synaptic transmission, we found that there was a selective decrease in the frequency of spontaneous miniature EPSCs as well as in the evoked EPSC, suggesting that there was a decrease in synaptic strength. That was somewhat surprising because this is a heterozygous mutation. When we perform similar experiments in mice, we don't see that, suggesting that at least in this type of human neuron that we are creating with these from ES cells, there is actually a significant effect of just a heterozygous deletion on the synaptic strength. It turns out that this is not a global change in synaptic strength, but rather a change in the regulation of synaptic transmission, because when you do stimulus trains as shown here, only the very initial responses are abnormal, as demonstrated with the first error, whereas as you can see with the second error in this plot that shows absolute EPSC amplitudes, it normalizes. In other words, it's a dysregulation of the synaptic response. It's not that the synapses are simply uniformly decreased in strength. So this result as an exemplary experiment validates the fact that Neurexin 1 mutations are meaningful, at least in human neurons, and that one can approach analysis of these mutations in such a way. However, the limitations of this approach are obvious in that you can't really analyze anything more sophisticated with human neurons in a dish. You can make different types of neurons, but that's about it. You can never actually look at the same diversity of properties that neurons and synapses normally assume in a real brain, let alone at circuits. So we do need mouse models in parallel to actually identify the functions of these various genes that we are studying, as well as the results of the mutations in these genes. And so what I want to do in the next part of my talk is describe to you exemplarily our work on Neurexin 3, because that's the most advanced, as a way of trying to communicate to you how diverse and multifaceted the functions of these proteins are, and in many ways, at least for me, unexpected. Neurexin 3 is not as frequently implicated in neuropsychiatric disorders, but it is sometimes more rarely mutated. So this is also relevant for neuropsychiatric diseases. The starting point of these experiments that I'm going to describe to you is the generation of conditional Neurexin 3 alpha beta knockout mice that abolish all expression of Neurexin 3 isoforms, and that had to be conditional because the constitutive knockout mice are lethal. When we analyzed these conditional Neurexin 3 alpha beta knockout mice in cultured neurons, hippocampal cultures in a dish, we found that they exhibited a significant but surprising phenotype right from the very beginning. What you see here are electrophysiological measurements of synaptic transmission in these cultured neurons that were infected with lentiviruses expressing either mutant CRE as a control or active CRE to delete Neurexin 3 alpha beta. And what you can see is that when you look at AMPA receptor-mediated EPSCs, there's a massive decrease in amplitude. However, NMDA receptor-mediated EPSCs in the same cultures are normal, as are iPSCs. This is surprising because Neurexins are supposedly presynaptic. A presynaptic phenotype would manifest in an equal decrease of AMPA and NMDA receptor-mediated responses that are both mediated by glutamate that's being released. This is a classically postsynaptic phenotype, and indeed, when we looked at minis that I'm not going to show you, we also had a decrease in mini amplitude, which is a classical postsynaptic phenotype. So we wondered whether there may be a change in postsynaptic AMPA receptor levels, and in order to test this, we went right directly towards measuring surface AMPA receptors at synapses using immunocytochemistry, as shown here. We used the same cultured hippocampal neurons, fixed them without permeabilization, and looked at the surface exposure of glutamate receptors as well afterwards, after permeabilization, at the levels of intracellular synaptic proteins. And what you can see here is that the density of puncta, synaptic puncta, is unchanged, as shown here for glutamate, GLU-A1, which is an AMPA receptor, PSD95, which is a postsynaptic scaffolding protein of excitatory synapses, and V-GLUT1, which is a presynaptic excitatory synapse-specific protein. However, the size of the puncta, which corresponds to a number or levels of AMPA receptors, is decreased in the Neurexin 3 alpha condition, knockout condition, to the same extent as the EPSC amplitude, demonstrating that indeed there's a loss of AMPA receptors from the surface. There is a trend for PSD95 that's not significant. V-GLUT1 is positive. The decrease in AMPA receptors on the surface is not due to a decrease in AMPA receptors levels in the total cell, because quantification of these AMPA receptors by immunoblotting, as shown on the right, shows that they are normal. So what is why are these AMPA receptors lost when you delete Neurexin 3 alpha beta? They are lost because there's too much endocytosis. Without going into the details here for reasons of time, let me just show you that quantifications show that AMPA receptor internalization, as shown on the right, is significantly increased. So in other words, what we have here is deletion of one set of neurexins, Neurexin 3 alpha beta, presumably presynaptic, leads to a destabilization of postsynaptic AMPA receptors in cultured neurons. We wondered if this is a developmentally fixed phenotype and the general function, whether this reflects the general function of neurexins. And so we tested whether this phenotype could be rescued by re-expression of different neurexins. And what we found is, exemplarily, when we re-express Neurexin 3 alpha with outer splice insert and splice at number 4, that this was fully rescued, as shown here, as it was by re-expression of Neurexin 3 beta with splice site number 4 out. So it could be fully rescued. It wasn't developmentally fixed. However, when we try to rescue with the same Neurexins but with an insert in splice site number 4, it could not be rescued, suggesting the possibility that this phenotype depended on a certain pattern of alternative splicing of Neurexin 3. Since this site of alternative splicing, splice site number 4, as I showed you earlier, I'm sure you remember, is extracellular, this suggested that this action of Neurexin 3 is mediated by an extracellular interaction of Neurexin 3. So we made a variant of Neurexin 3 where Neurexin 3 is exposed on the cell surface via a GPI anchor, and we tested whether that could rescue the phenotype, and as you can see here, it's fully rescued. So you don't need a transmembrane region, you don't need a cytoplasmic sequence. However, if you do the same experiment with a secreted variant of Neurexin 3 that is actually encoded by a normally occurring splice variant, it can't rescue. So it needs to be anchored on the plasma membrane in order to function. Now these results intrigued us because alternative splicing of Neurexins from the very beginning had been something that we were very interested in as a possible way of encoding information, of basically forming a recognition molecule. So this was the first evidence that alternative splicing may be physiologically important. And so we wanted to test whether alternative splicing actually is physiologically important in a way that wouldn't involve rescue experiments, because rescue experiments are always, no matter what people tell you, overexpression experiments. And overexpression experiments, at least in neurons, have enormous numbers of problems. So we wanted to do this in a way that would otherwise be as normal as possible. We didn't just want to overexpress different types of neurexins and see what the effects were. And so we generated mice, mutant mice, that contain a mutation in the splice acceptor sequence of the exon that is alternatively spliced in this event of alternative splicing. Specifically, what you see here is the wild-type sequence. You can see that that splice acceptor sequence is abnormal. It contains all these A's that are not supposed to be there. We converted this into a canonical splice acceptor sequence, as shown here in red. The idea here was that the canonical splice acceptor sequence would be constitutively spliced in, that this exon would no longer be spliced out. And indeed, that worked. It is constitutively spliced in. It's totally now becomes from alternatively spliced to non-alternatively spliced. In addition, we flanked the exon that is alternatively spliced here by LOXP sites in order to simply delete it constitutively. This way, we could genetically manipulate expression of either Neurexin 3 always containing or lacking sites of alternative splicing. As a result, when we have this mutant in control conditions, the mutant control, the sort of where we will always have splice site 4 plus. If we add Cre, we will have splice site 4 minus. When we analyze these neurons using the same approach that I already showed you, cultured hippocampal neurons, now in comparison to an unrelated wild type, we found that again, the EPSCs in the mutant condition with the SS4 plus were decreased about 40 to 50%, both compared to the wild type and compared to the SS4 minus. This was the same phenotype as the Neurexin 3 alpha deletion. No change in NMDA receptors, no change in iPSCs. So it seems like indeed the rescue experiments were correct. This was totally dependent on alternative splicing. Moreover, what we found is that this phenotype could be rescued by all SS4 minus neurexins, but not SS4 plus neurexins, and that this phenotype was also due to a change in AMPA receptor endocytosis. We then tested other phenotypes and found that they're exactly the same, suggesting that the entire loss of function phenotype of Neurexin 3 alpha beta was dependent on the expression of SS4 minus Neurexin 3 alpha beta. And this raised a number of questions. One is whether this whole set of phenotypes actually corresponds to something that happens not only in the cultured neurons but in vivo. Another one is whether Neurexin 3 here acts actually as a presynaptic molecule, since the phenotype is so specifically postsynaptic. And so we used an in vivo approach to test this. We took the mice that contained the conditional mutations, either the knock-in or the knockout, and we injected stereotactically AAVs expressing either control mutant Cre or active Cre recombinase into the CA1 region, as shown here in these sections. We then cut slices 2 weeks later, and we analyzed these mice electrophysiologically by measuring the effect of these presynaptic mutations here. This thing sort of doesn't allow you to click twice. You have to wait. It's kind of a slow down event. Yeah, maybe that's a good thing. Anyway, so by recording from subiculum neurons, which are not infected, so only presynaptic neurons are infected. Postsynaptic neurons are not. Now it comes back. So postsynaptic neurons are not. These are the subiculum neurons that are recorded from. These are the infected neurons. Here are the stimulating electrodes. And this allows you then to analyze specifically presynaptic effects. A complicating factor of these recordings is, however, that the postsynaptic subiculum pyramidal neurons are composed of two broad classes of neurons. These classes of neurons have different intrinsic electrical properties and different forms of synaptic plasticity. Specifically, they are regular firing subicular neurons that fire regularly, and there are burst firing subiculum neurons that fire in bursts when you depolarize them. Moreover, the regular firing subiculum neurons have classical postsynaptic LTP that's NMDA receptor dependent. The burst firing neurons have presynaptic LTP. So in other words, you have to do twice the work because you have to analyze both of these separately. Nevertheless, this preparation allows you, because of this properties, not only the analysis of specifically presynaptic manipulations, but also the analysis of different forms of synaptic plasticity. What we found in these experiments is that both for the Neurexin 3 alpha beta deletion and for the knock-in, there was a loss of AMPA receptor mediated responses. What you see here is a comparison for the knock-in of the wild type with the knock-in and the presynaptic excision of splice site 4. And you can see that the phenotype is exactly the same as what we observed in cultured neurons. This is true for both burst and regular firing subiculum neurons. So this validates the culture experiments and demonstrates, moreover, that it has to be presynaptic because the only manipulation here is presynaptic. Moreover, and somewhat surprisingly, we found that these manipulations led to a change in LTP. What you see here are measurements of LTP as demonstrated in this diagram on the left. And when you quantify this as the extent of LTP, you can see that in the control knock-in condition, which is SS4 plus positive, insert always in, there's an almost complete loss of LTP, which can be rescued by presynaptic excision of the splice site number 4. This was surprising because it basically demonstrates that postsynaptic LTP is dependent on a presynaptic gating signal that tells the postsynaptic neuron whether it is allowed to actually undergo this type of AMPA receptor trafficking in a yes or no manner. Now, the one issue that arises here is this the only function of Neurexin 3? Is that what Neurexin 3 does? And if so, why is there a splice site 4 plus at all? Just to turn it off? Or are there other functions that we don't see in this particular neurons because they represent one type of neuron and there's many different types of neurons in the brain? And so to address this, we went to a totally different type of preparation. We started to analyze similar processes in the olfactory bulb. And I'm only going to tell you about one particular experiment here to illustrate this, because what we found is that there the situation was completely different. What you see here as an illustration is the culture of olfactory bulb neurons, which consist largely of only two types of neurons, namely mitral tufted cells, which are big and are shown here in the middle, MT, and granulate cells, which are more numerous, small, and inhibitory. And so you can analyze synaptic transmission either in culture or in slices using from these knockout mice or knock-in mice. And what we found is that, now switching back to the Neurexin 3 alpha beta conditional knockout, that the phenotype was totally different. AMPA receptor-mediated responses were normal, NMDA receptor-mediated responses were normal, but iPSCs were now decreased more than 50%. So now there was a loss of synaptic transmission in a different type of synapse. Moreover, when we analyzed the nature of this impairment in synaptic strength, it was not a postsynaptic phenotype. It was a classical presynaptic release phenotype with a change in paired pulse ratios and a change in release probability. Finally, this phenotype turned out to be independent of alternative splicing of splice site number 4. We could rescue it, as shown here, with SS4+, as in fact, under control conditions, just overexpressing in this rescue experiment SS4+, in wild-type neurons led to an overshoot of the AMPA receptor of the iPSCs. Moreover, this phenotype could not be rescued by GPI-anchored neurexin that's expressed on the cell surface, different from the AMPA receptor phenotype. So completely different. Molecular mechanisms are different, phenotypes are different, nothing in comparison. Different types of neurexins, different mechanisms. So what I've told you in this part of my discussion with you is that Neurexin 3, as an exemplary neurexin, has different functions in hippocampal versus olfactory bulb neurons as exemplary brain regions. In the hippocampus, presynaptic neurexin controls postsynaptic AMPA receptor levels. This is mediated by an extracellular interaction with postsynaptic neurexin ligands and is also involved in controlling classical postsynaptic NMDA receptor-dependent LTP, which also involves AMPA receptor trafficking. Moreover, this function of Neurexin 3 is regulated tightly in an all-or-none manner by alternative splicing at one particular site, splice site number 4. In contrast, in olfactory bulb, you have an essential function of Neurexin 3 in controlling or regulating or enabling release from inhibitory neurons in a manner that involves the intracellular sequences of Neurexin 3 and that is independent of splice site number 4. And to try to sort of visualize this, to try to understand this better, let me show you our atomic high-resolution model of Neurexin 3, which you can see here. Something you probably all need in the woods around here. Basically, this is Neurexin 3, and you can see there are all kinds of things coming out of it. And the way we envision this is that this is basically a platform. It's a scaffold, if you want, that can mediate very different functions depending on the context. For example, if you like wine and AMPA receptors, here you are. Or if you want to cut something, NMDA receptor-dependent LTP, presynaptic release probability. Neurexins are also essential for endocannabinoid, regulation of endocannabinoids, mediated modulation of synaptic transmission, which I haven't discussed, and presumably other functions as well. And we believe that neurexins have different functions, especially Neurexin 3, as shown here, in different types of synapses of neurons because of the context in which they're expressed. Possibly other neurexins that are differentially co-expressed, possibly other as yet incompletely understood molecular pathways. I hope that with this discussion you get a feeling for how nonlinear in many ways even molecular events can be. So you have one molecule, alternative splicing, different context, completely different manifestations, demonstrating that we really need to actually dissect individually synapses and molecules in order to understand what's going on in particular information processing events in the brain. Let me now, in my final section of my talk, and I'll rush through there, to talk about a totally different component of our work, something that relates to the behavioral consequences of mutations in neurexins and their ligands. And let me talk to you about one particular mutation in neuroligin 3 because that is where we had the best insights at this point. As a reminder, neuroligin 3 is a postsynaptic ligand of neurexins. It's an X-linked gene. There have been many multiple point mutations described in patients that are associated with autism. In addition, there have been multiple large-scale deletions that have been associated with autism. So just like neurexin 1 in particular, this molecule is associated with autism. Some time ago, we set out on a strategy on trying to understand how these mutations might actually predispose to autism, might cause behavioral abnormalities, and what their effects might be on synaptic transmission. And in order to do that, we genocopied 2 different mutations in neuroligin 3 that had been described for patients in mice. One was just a deletion, a knockout. Another one was a knock-in that recreates one particular point mutation, R451C. We analyzed comparatively their phenotypes, and to our surprise, in the initial experiments, we could not find anything that these 2 different mutations had in common. That was very confusing, not to say depressing, because you have one gene, 2 different mutations in humans associated with basically the same disorder. Nevertheless, in a mouse, they seem to cause totally different impairments. And this was puzzling and actually set us back for some time. Now, what we also observed in these experiments is that the mutation, the R451C knock-in mutation, had a much more severe phenotype, a much more diverse phenotype than the knockout. And that mutation, in addition to causing diverse phenotypes, also caused a destabilization of the neuroligin 3 protein. So it was both a knock-in change in the sequence and a loss of function, since more than 90% of the protein was gone. You had 10% that was abnormal, 90% was gone. And so this led to the idea that the R451C mutation, which gave such a diverse panoply of phenotypes, might cause gain of function in addition to an underlying loss of function that is caused by the neuroligin 3 knockout. And so, in particular, what we observed in these early experiments, as did many other laboratories, is that the behavioral phenotype of these mice was very different. In the water maze memory test, one of the R451C was doing better than what controls, whereas the knockout has no phenotype. Social approach was decreased in the R451C, but social memory was selectively decreased in the knockout. And so we were wondering what this might mean. And in considering this, I would like to remind you that when we use mice as a sort of model for trying to analyze, study any kind of disorder, certainly for autism, this is always meant to be in a way that a behavioral impairment is a proxy for a change. There is no way a mouse will ever behaviorally model a human condition. I mean, that's just impossible. In fact, a lot of bad things are said about mouse models as a way of analyzing human diseases. I think they're still great models, despite the fact that almost always the phenotypes are different. I think that the phenotypes that I've observed can still be used as proxies, but we can discuss this later. Okay, so what we noted when we considered this situation about neuroligin 3 is that several other mouse models that were supposed to model autism had one particular phenotype, which consisted in an increased ability to do a repetitive motor learning task, which is the Rollout Assay. And this assay consists of simply putting a mouse onto a rotating rod, as shown here on the top left, and then measuring as the rod accelerates how long it takes for the mouse to fall off. And then you do this several days, 3 trials a day. In our protocol, 2 days at 4 to 40 RPM, and then 2 days at 8 to 80 RPM. And what you can measure with the time that it falls off, you can measure the initial time it takes for the mouse to fall off as the ability to coordinate movements, and the learning as the ability to increase their staying on the rod. And what we found, confirming other data in the literature, is that the alpha-5-1c mice were better at this. They basically they learned better to stay on the rod compared to littermate wild-type controls. And the way how you can analyze this is to put a linear line through this curve, and the slope of this curve gives you the learning rate, and the intersection with the y-axis gives you the initial coordination, and then you can plot this, and you can see initial coordination in the mice is fine, but the mutant has an ability to learn better. So this has, as a phenotype to analyze, a lot of advantages, because if a mouse can do something better, it's always more specific than if a mouse can do something not as good. An impairment in an ability, in a behavioral ability, can be caused by thousands of changes, presumably. An increase in the ability is likely to be more specific. So the first question is this shared by the knockout? And of course it is, otherwise I wouldn't talk about it. It has exactly the same phenotype. Okay? So here we have then a phenotype that we can use potentially as a proxy for an autism-related impairment. Now the question obviously arises whether this is really an autism-related impairment, and I don't know that. But we can at least hypothesize that there's some relation in that this behavior does in some way distantly resemble stereotypic behaviors. For example, one experiment that we did in this regard is to actually measure the movements of the mouse as it was learning the task by using videotaping of their feet, as you can see here on the left. And when you do this, you can measure how consistently they put their movements, that is, how stereotypically they actually move around. And you can see that the mutant mice learn much, much better how to move the step location. They decrease the standard deviation, that is the variability of the step location. They decrease the standard of the step length and of the step timing, demonstrating that these mice actually are better on these tasks because they are better at learning to do the same thing over and over again. And that in some way is coordination is a stereotypic movement. It's a learning of a stereotypic movement. So what we have then is diverse behavioral phenotypes of these two different kinds of mice in some aspects, but similar phenotypes in the low motor learning and a stereotypic motor learning. In other words, we have a gene, a molecule, we have a behavior, and the question then is, what's in between? What are the synapses? What are the cells? What are the circuits? And this is what we wanted to explore. And in order to do that, we needed conditional knockout mice, which we generated using standard approaches. And then we analyzed these conditional knockout mice to specifically ask which brain region is involved here. And to do this, we focused on the cerebellum or striatum, because these are the two brain regions that have been associated with motor learning in the literature. They are known to be involved in motor learning. So we crossed these conditional knockout mice first with a mouse that expresses Cre recombinase in Purkinje cells, which is the only output, postsynaptic output neuron of the cerebellar cortex. And when we did this and analyzed the resulting mice for their rotor rod behavior, we found that they had no phenotype in terms of the rotor rod learning, even though they did have a phenotype just like the knockout, the constitutive knock-in knockout mice in terms of hyperactivity, which I haven't mentioned before. So the cerebellum is clearly not responsible. So then we went to the striatum, and as you probably all know, 90% of striatal neurons are made up of inhibitory projection neurons called D1 and D2 MSNs, medium spiny neurons, that can be separately genetically manipulated. So we first crossed these conditional neuroligin 3 knockout mice to Cre expressing mice that express Cre in one particular medium spiny neurons, the D1 neurons. And when we did this, we got a complete recapitulation of the phenotype. In other words, the D1 MSN knockout caused the same increased motor learning ability and also hyperactivity as either the knock-in or the knockout mouse in the entire brain. And this suggested to us that the striatum is involved. When we looked at D2 neurons using a different Cree line, we found no phenotype. So only D1. So this now localizes this defect to one particular type of neuron, which is gratifying because it gives us a clue. But it also raises other questions. One is, the striatum is huge. In fact, recent studies have shown that all of the information that goes through the cortex always also goes through the striatum and actually the cerebellum in loops. And so in other words, whenever something happens in the cortex, it also happens in the striatum, which is enormous, and the cerebellar cortex, which is also enormous. So the question thus arises, which part of the striatum? Another question that we were very interested in is whether this phenotype is something that is caused by a developmental abnormality. So if the neurons don't develop right, and once that's done, you can't change it. And so these are the two questions we wanted to address. And in order to address both of them at the same time, we used clear injections using viruses, AAVs in this case, that express large regions, large areas of the striatum by stereotactic injections, either of the nucleus accumbens or of the dorsal striatum. And when we did this, nucleus accumbens injections reproduced the phenotype, suggesting that the ventral striatum, the nucleus accumbens, is essential. When we did the same thing for the dorsal striatum, no phenotype. So within the striatum, it's the ventral striatum. What these experiments also suggest is that this phenotype can be induced in young adult mice after development. So it's not necessarily developmentally conditioned. It seems to be a phenotype that depends on the continued function of the proteins in the striatal neuron, since it can be reversed by deletion. So we wondered if we could also rescue the phenotype by re-expression after development of neuroligin 3. And we did the same strategy in terms of using a Cree-dependent D1 specific strategy of re-expressing neuroligin 3. And just to show you it's fully rescued, the phenotype goes away. So this is not a developmental phenotype. It's a phenotype that depends throughout life of the mouse on the continued expression of neuroligin 3. So for those of you who are familiar with the striatum, obviously the question arises, why the nucleus accumbens? The dorsal striatum is the one that's supposed to do motor learning. The ventral striatum is supposed to be involved in reward and addiction, all kinds of stuff. In fact, the ventral striatum right now is probably the most studied brain region because of all these implications in these very interesting behaviors. So the most immediate possibility here that arises is that this phenotype in the ventral striatum is due to a fact that the mice simply want to stay on the rotor rod longer. In other words, that they have more motivation. That it has nothing to do with actual motor learning. I can't exclude this possibility completely, but we did perform some studies on the reward circuits, on the reward behaviors using cocaine-conditioned place preference. And as you can see in these experiments, and I'm not going to go into them because of the reasons of time, there was no phenotype. So at least there is no obvious massive change in reward behaviors. So then finally, we wanted to know whether this change in behavior is associated with the change in synaptic transmission, and what kind of synaptic transmission. To our disappointment, we found no change in excitatory synapses. Neuroligin 3 is postsynaptic. Most of the inputs into MSNs are excitatory from the cortex or thalamus, but there was no change. That was surprising to us, but we did observe a change in inhibitory synaptic transmission. This is illustrated here in that in D1 MSNs, the knockout causes a decrease in the frequency but not amplitude of many iPSCs consistent with the change in the number or strength of inhibitory synapses, there was no change in D2 MSNs. We then analyzed this in more detail by measuring in the same neurons both excitatory and inhibitory transmission to see if there was a change in the balance of excitatory versus inhibitory inputs. And indeed, when we do such experiments in D1 neurons, we see a massive decrease in the inhibition-excitation ratio, whereas in non-D1, i.e. D2 neurons, no such change was observed. So there is a specific synaptic change that that correlates with the behavioral impairment. The same synaptic changes observed both in the knockout and in the knock-in, demonstrating that, again, in this particular neuron, in this particular circuit, these mutations have the same loss of function phenotype. So what the studies as a whole thus demonstrate is that the neuroligin 3 loss of function causes one discrete change that seems to be shared by all neuroligin 3 mutation in behaviors. That is a gain of function in terms of a stereotypic motor learning behavior that can be localized to a specific type of neuron and a specific brain region and a specific type of synapse, although we do not know which subtype, if you want, of inhibitory synapse is involved. This impairment is not developmentally fixed. It's not a developmental phenotype. So at least for this particular set of experiments, we could demonstrate that these classically neurodevelopmental disorders may not be that neurodevelopmental, but may be basically continuously operating disorders, which is obviously important if you consider any possible future therapeutic interventions. Okay, so I'm done. 5 minutes over, sorry. I hope when you walk out of here, you'll never forget Neurexins and Neuraligands, or me. I'd be happy to put this to discuss the implications of these findings for our understanding not only of the brain but also neuropsychiatric disorders. In anticipation of such a discussion, I want to just show you one sort of idea that I would like to illustrate with this cover from the New Yorker a couple of years ago to try to give you a feeling for how we think about these things. What this cover shows was just after Christmas, and it shows children putting on a play and their parents videotaping the play. Happens if you're a parent, that's what you do, right? But what's interesting here is that the parents actually don't videotape the play. They videotape their kids. Okay? So they don't actually pay any attention to the play. They only videotape their kids. If you had those videotapes separately, could you put back together the play? Okay? And that's basically the situation we face when we try to reconstruct how something works. You need to actually have a videotape of each individual player or each individual protein or gene or whatever, but you also need to put them together. Moreover, in cases where you have mutations, that is, let's assume there's an actor missing, but the play would go ahead nevertheless. And obviously the other child actors would react to the absence of their friend or colleague. And you would also videotape this. That's probably the situation we face in neuropsychiatric and other disorders. Could you also reconstruct it, and could you sort of think about ways of making up for the absence of that particular player? And so I think that we need to actually understand the players, that is individual molecules, genes, but we also need to understand, put it together and try to relate it to what happens when these things are absent. Let me close by acknowledging the most important people for this particular work. The Neurexin 3 studies were done by Jason Aodo, who is now has his own lab at the University of Colorado. Chang-Hee Pak, Yingxia Zhang, and Tamas Danko did together the human neuron work that extends beyond what I discussed. And finally, Neurexins story was done by Patrick Rothwell and Mark Fucillo, who are both now have their own labs at the University of Pennsylvania and Minnesota, respectively. I have a lot of collaborators who I love, including Marius Wernick for the stem cells, Axel Brünger for the structural biology, and Rob Malenka for slice physiology. Thank you very much for your attention. If people have the time.
Dr. Südhof, we take a couple of questions. Leslie.
Hi, thank you very much for a wonderful talk. I'm wondering if you and your lab or Rob have looked at the physiology in the NL3 mutants in the CA2 region and what you see, and if you manipulate NL3 in that region, if you look back at the deficits you see in the social behaviors.
Great question. No, we have not looked at CA2. So we only, guess that's lab tradition. So we should probably pay some attention to CA2. We haven't traditionally done that. You know, we do CA1, we do CA3, we do dentators, but
It's just between CA1 and CA2.
it is. I'm sorry.
A little bit extrapolation, but with the Swiss Army knife, I was thinking about the issue of redundancy in different aspects of the brain and how, you know, even isolating one particular small area is sometimes perhaps confusing to the overall picture. And if you could comment on that. The other question is, with the rotarod mice, were they also, those mice specifically, tested for the other for instance, hyperactivity or other social behaviors, or was that done in mice that were not rotarod trained?
So we have independently tested hyperactivity. In fact, hyperactivity is the first thing we always test. It always is present in these mutations. It's present with the D1 knockout of neuroligin 3. It's present with the cerebellar knockout, which, however, does not have the rotarod phenotype. We have done previous analysis, extensive analysis of neuroligin 3 knockout and knock-in behavioral phenotypes. We find all kinds of things, but I haven't gone into this, but we didn't redo those extensive analyses with these conditional localized studies. So with those, we have not done those. We have only done those two that I discussed here. Obviously, behavioral experiments in mice have an unbelievable number of caveats associated with them, including how you do the sequence of these assays. Yeah. That was one of the reasons why we basically first do activity, and then we go into the rotarod, and then we sacrifice the mice and study whether the manipulations were actually effective, whether they were the right thing. Yeah, and obviously everything is done blinded with littermates, no question. Yeah. As regards redundancy, redundancy is an unbelievably important issue because it's much more complex than it sounds. It's complex for a number of reasons. One is there is different types of redundancy. There's compensation that happens developmentally in some synapses, but in our cases, we have observed very rarely. There's parallel pathways that are mediated by homologs. There's parallel pathways that are mediated not by homologs. As a result, any kind of genetic manipulation that results in that you're analyzing for a particular phenotype and doesn't have a phenotype in this particular assay, it doesn't mean that manipulation, that the manipulated gene has no function in that assay. It just means that there's no phenotype. That's basically what it means. and so one always has to be aware of that. And I refer to this as phenotypic bottlenecks. So I think that many molecules are likely, genes are likely redundant in brain. In different brain, they're differentially redundant in different neurons, types of neurons, different synapses. And I call this the phenotypic bottleneck because there are phenotypes that arise from manipulations, and these phenotypes are due to these bottlenecks, which represent neurons which, for whatever reason, don't have a particular redundancy that's present in other neurons. And that's why I call it a bottleneck. And yeah, I mean, we can never say what something doesn't do.
That was a great talk. I guess, seeing the structure of Neurexin in such detail, one thing that I would note is if you're trying to open a bottle of wine, it's very hard to then clip your nails with the scissors, for example. So this is
sequence is important, right? Your first clip.
So I guess kind of my question was, since you see these two different behaviors of the molecule in these different synaptic contexts, can you know about what type of molecules might dominate, say, the corkscrew function over the scissors, or based on the connections, and how Neurexin will act?
Well, if I understand your question correctly, what are the sort of the mechanistic basis for these various things? Yeah, I don't know. There are several ligands that Neurexins bind to. Presumably these are important. I would love to know, and we're trying to analyze some of these things, but at this point, I just simply don't know.
So kind of along that same vein, you've now really proven that the different splice variants can sort of create a code that would regulate the properties of specific types of synapses. But now you're faced with the problem that you have thousands of splice variants and diversity of synaptic function. And how do you crack the code to figure out which types, the entire code?
You make it sound very attractive. One at a time, yeah. We'll get there eventually.
You crack the code by drinking a wine first.
Yeah.
Well, thank you very, very much.
Thank you.